you so much for the kind introduction. As mentioned, my name is Chloe, and today I'll be talking about our recently published work titled Autism-related Dietary Preferences Mediate Autism-Gut Microbiome Associations. So I think most of us think of autism in relationship to social and communication difficulties, but the other diagnostic criteria also include restricted and repetitive traits, as well as unusual sensory sensitivity. There's been increasing interest between autism and the gut microbiome, and that's motivated by a pretty strong body of evidence that gut conditions are more common among kids on the spectrum. And so then this led to some observational human studies, which were overall small, which found some associations, albeit inconsistent. This in turn progressed to animal studies, suggesting maybe an element of causality, and even further to early open-label fecal microbiota transplant clinical trials. This has also been sort of occurring at the same time as an explosion of sort of the industry interest in autism and the gut microbiome, and the proliferation of so-called therapies that claim to use to somehow leverage the microbiome to alter autistic traits in some ways, and in turn has led to families bringing these therapies to their clinicians and asking questions about them. However, the field has so far been limited by studies of small sample size, inadequate correction for multiple testing and confounders, and in fact, there are almost double the number of reviews on the topic as there are primary literature. So we performed the largest autism metagenomic study to date, leveraging the Australian Autism Biobank and QTAB cohorts. This is a snapshot of the study design, which included a total of 250 kids, each with deep phenotyping, so psychometric, diet, and stool consistency data, as well as SNP genotyping. And this is showing a sibling comparison of 50 pairs nested within this autism, not autistic, unrelated comparison, unrelated children. And we were quite privileged to work with Microba, who gave some very high resolution insights into the microbiome using their large database and profiler. This is a snapshot of the group demographics, so the age range from 2 to 17. Unfortunately, there was a bias towards males, but this is entirely expected because boys are more likely to be diagnosed with autism than girls. So just to kind of talk about the data quickly, in terms of the metagenomics data, after cleaning, there were 607 bacterial species and 2 million genes, these coming from the Microba profiler I mentioned earlier. In terms of phenotypes, we got dietary data from the Australian Eating Survey, and I'll talk about that more in the next slide. Stool consistency, as well as psychometric and demographic data on autistic traits, cognitive performance, and as I mentioned with the SNP genotyping, using that to generate polygenic scores. So just to talk quickly about the dietary data, we looked at it in two ways. So firstly, just the raw variables, so the frequency of consumption of over 120 food level items, and then derived variables. And the derived variable that we focused on was the percentage of energy contributed by each of these food groups, which can be broadly broken down into healthy or core food groups in green and non-core or unhealthy food groups. And so we got these kind of food groups adjusted for energy intake, and then sort of used principal component analysis to identify dietary profiles, which is shown here. So PC1, dietary profile for a plant-based healthy diet, this one for dairy, and this one for high in meat. So there's many ways in which we could investigate a potential association between autism and the microbiome. And the first question we looked at was whether the microbiome as a whole is associated with a trait, for example, autism diagnosis. And we specifically interrogated this question by asking what proportion of variance in a trait can be explained by the microbiome as a whole. And when I refer to the microbiome as a whole, we looked at that in different ways as well. So in terms of the common species, the rare species, the common genes, and the rare genes. And this OREML approach gives us a measurement between 0 and 1, where 0 suggests no association, and 1 is that there is a very, very strong association. And these results can also be interpreted as representing the upper limit of predictability. And so we first looked at age as a benchmarking trait, finding that the species level estimate, as shown here, is consistent with previous estimates, as shown by this blue line. But it was interesting that the gene level data explained almost all of the variance in age. In stark contrast, there was negligible association between any of these microbiome measures and autism diagnosis, but there was a significant association for diet. We also looked at stool consistency and this dietary profile, as these are intuitively associated with the microbiome, and we reassuringly found some strong associations there. So that didn't kind of look so good, and so we looked at some other ways in which there could be an autism microbiome association. We first looked for differential abundance of individual species. We found one significantly less abundant species in autism, which was Rhambutia tiemanensis, which was interesting because it belongs to the Clostridiales order, which has previously been associated with autism diagnosis in 16S studies. Overall, though, there wasn't really much evidence for there being many species, individual species, being associated with autism diagnosis. Out of interest, we also zoomed in on the genes that were encoded within the Rhambutia tiemanensis genome and performed a differential abundance analysis and did find some associations with autism diagnosis there. We didn't find any association with either alpha or beta diversity and autism diagnosis, so no difference in alpha diversity and no difference in beta diversity. No difference in stool consistency. But interestingly, again, there was this association with diet, with the autism group seeming to consume less meat. And that's shown in this plot. So this is dietary PC3 representing less meat. And this is showing the coefficients of a linear model where the Y variable is the dietary profile. And here we have the group variables. So there is a significant difference there. Overall, though, I think this evidence that I've shown so far suggested that there was negligible direct association between autism and the gut microbiome, but there was sort of this smoking gun signal relating to diet. And so we went back to one of the core diagnostic criteria for autism diagnosis, which includes restricted and repetitive behaviors and interests. And we hypothesized that these behavioral differences and preferences may manifest as restricted diet, and that this in turn may be associated with the microbiome. And to, I guess, assess or estimate restricted diet, we just use Shannon diversity on the food level data matrix. And we did find that autism diagnosis was associated with reduced dietary diversity, despite having greater variance as well. As I mentioned before, species diversity, alpha diversity, was not associated with autism diagnosis, as shown here. But there was a significant positive correlation with dietary diversity, suggesting that it may be a downstream process. And then we went a bit further and asked whether there may be differences in stool consistency, and did find evidence linking lower species diversity and runnier stool, as shown here. Furthermore, these associations were all robust to covariates. So this is finding 1 with covariates here, finding 2 with covariates, and finding 3 with covariates. We then wanted to drill down further and ask whether there were specific aspects of autistic traits that may be associated with restricted diet. And so we first looked at continuous measures of the autism spectrum. And we used both clinical measures, so psychometric measures, as well as genetic measures as measured by this polygenic score, which captures the polygenic, the common variant contribution to autism, and found that there was a significant association between these measures and restricted diet. Similarly, with restricted and repetitive traits, as well as some evidence of sensory preferences differences with restricted diet. And these associations are interesting because we only had data for about 100 kids out of the full 250. So this is relatively smaller sample size. We do caveat that this is a small data set and these associations aren't, blowing your socks off strong, but overall we think that they are consistent with this causal model that we propose. So in conclusion, we found that direct microbiome autism relationships are weak compared to other traits, but it looks like autistic traits and preferences restrict diet, and that these in turn drive microbiome differences. In terms of implications, I think it's important to temper this hype around the autism microbiome and empower parents and clinicians to make informed decisions when there are so many microbiome so-called therapies floating around, suggesting that there may be making a difference to these kids when they may not and are expensive. We also highlight the importance of adequate nutrition for kids on the spectrum. And finally, we, urge the broader field to look at the impact of behavior and diet and microbiome causality, not just for psychiatric traits, but for any diagnosis in which there may be some associated difference in dietary preferences. So this is just where you can read more about our work. And I'd just like to thank the huge team involved with this project. Thank you so much.